Hello, ladies and gentlemen. It's my pleasure to bring to you the first video of this series where we are dealing with the application of engineering sense to the biological intelligence mechanisms. And the first in this one is, of course, the cytoskeleton, the executor of intracellular intelligence. That's the article for today. And yes, so this was the series that we had announced some time back. So today we are on the first video dealing with cytoskeleton and the intracellular intelligence. Okay. Now this is how a biological cell would look like typically. Of course, there are a lot of variations from cell to cell, but then typically, yes, this is a representative biological cell showing all the possible features which could be there, right? Now, so there is an outer envelope which is flexible, but which is largely impermeable, okay, in the sense that it isolates fluid on either side, okay, so that's the cell membrane. And similarly, there are various similar membranes inside which form the various vesicles, etc., within the cell, okay. So the cell is supposed to be filled with cytoplasm, and cytoplasm has two components. Cytoplasm is supposed to be a jelly-like fluid substance, but any fluid substance in a flexible envelope would always end up looking like a droplet, like a drop shape. Okay. But as you can see here, the cell has very elaborate external and internal shape and structure and orientation, which is maintained by the skeletal members within the cytoplasm. So cytosol is one factor which is there inside. That is a solution like component of cytoplasm where the smaller molecules particularly can diffuse through as if they diffuse in a solution in a fluid, right? And the other one, the other component of cytoplasm is cytoskeleton, which is having fibrils, fibers, assembly of fibers, something like ropes or mesh, and or even something like spider web maybe, and which makes sure and which constrains the inner organelles and whatever structures are there in certain positions with relative to each other. Okay, so one function of cytoskeleton is that it defines the shape and orientation of the cell. Okay, and there are various shapes and sizes of the cell, as we were saying some time back. Uh, More technically speaking, of course, there are apical cells, there are basal cells, there are star-shaped cells, stellar cells which look like amoeba or neuron. Okay, there are cells which rest on one base and they grow from the base. So those are basal cells, and there are pyramidal cells and so on. But obviously, cells do have an orientation which is known to the cell. They know which side surface, what is the functionality. Okay, so to that extent, yes, cell has a map, though it is slightly fluid, but still it is quite elaborate. And the map and the disposition of various elements within the cell is ensured by cytoskeleton. What else could do that? Obviously, I mean, organelles cannot be filled like in a jelly because all of them could settle on one side like a lump. Okay, so obviously there is something which is bringing some order and some discipline and some interrelation. Okay, so the shape and orientation of a cell is controlled by the cytoskeleton. Now, cytoskeleton is sometimes conspicuous, sometimes it is inconspicuous. Sometimes it becomes visible, sometimes it becomes invisible. Right? There are occasions when you can see it, there are occasions when you don't see the existence of cytoskeleton also. Right? But it is definitely there and it is carrying out many intelligent functions. Okay. Now, one intelligent function is, of course, the shape and orientation of the cell. Okay. Because if you talk of tissues, if you talk of, let us say, intestine cells, okay, they are absorbing food from one surface and they are passing whatever has been absorbed to the bloodstream on the opposite side. So obviously the cell knows which side is which, from where it has to ingest, from where it has to secrete, right? So it knows from where to excrete and where to secrete, where to ingest, right? So cells definitely know their orientation. And also within the cell, the various organelles that you see here, these cisternae, these Golgi bodies, these various vacuoles, mitochondria, and so many things which are there in certain shape. Now, without a cytoskeleton, can the cisternae look like this as if they are seen in this drawing? Okay, they are more or less concentric, and they are not intersecting the nucleus, and they are not intersecting each other. They are lying more or less parallel, right? Now, what is that which is bringing this discipline into the cytoplasm? Obviously, it is cytoskeleton. It has to be some ropes, some flexible pillars, some restrictions which are allowing organelles to stay somewhere and preventing them to drift somewhere else, right? So that is an important function. Placement and shape of organelles and cisternae. It is very obvious that the order is brought into place by cytoskeleton. Now, also the cell surfaces have some characterization. They have some protein globules which are floating on the surface, which can move on the surface, but they have certain characteristics. Like some of them can act like a non-return valve or two-way valve or just a gate. Okay, and opening and closing of the gates, opening and closing of the walls is obviously getting executed through cytoskeleton. Okay, communication to adjacent cells through the fibers which are running through from one cell to other cell. Okay, now if some message has to be passed, obviously it is going through the cytoskeleton to the adjacent cell. There is no other way it can. A jelly cannot do that in a specific focused directional manner, obviously, right? So characterization and actuation of cell surface elements, operation of walls which are there on the surfaces, yeah, all that channels are opened and closed as required by the biological intracellular process. Yeah, all that intelligent function is executed by the cytoskeleton. Okay, much like in a two-wheeler there used to be some wire, some cables. Like there was a cable which used to operate the accelerator. There was a cable which shifts the gears, right, and a clutch. Something like that. There has to be some ropes which have to do some string pulling literally and operate things as required, right? Now all these intelligent operations mechanisms are realized through cytoskeleton, because there is no other possibility of anything in any other way could be done, right? Then the formation of vacuoles, basically, if you remember high school days and amoeba we studied. Now amoeba has a variable shape and it has what is called as pseudopoda. It can do locomotion. It can move by changing its shape. Okay. Now, what could govern that change in shape so that it moves in certain direction in certain way? Right. Obviously, it is the cytoskeleton. The skeleton. Okay. Okay. The shape of a tent is defined by the ropes. Now, if you change the tension of the rope, the shapes do change. Something like that. So pseudopoda is actuated through change in cytoplasm through cytoskeleton. Also, when there is a food particle coming on the surface, on the outer surface, as here or here, okay. Now, on the surface, there are some protein globules which can identify the food with other particles. Okay. A cell obviously would not go on ingesting anything and everything that comes on the surface. That would be too inefficient. So biological systems are obviously much more optimized than engineering system. So that optimization is brought through by actuation of some sensors on the surface, now which could be realized through the protein globules which are residing on the surface, and they give some signal to the cytoskeleton, and then cytoskeleton alters the shape and tries to form vacuole, it tries to form a vesicle which breaks off from the outer membrane and moves inside. Okay, something like this one here. Okay, now this is where it is forming on the surface, and this is where it is moving inside. Okay, and it has engulfed the food particle within the identified food particle which it is carrying inside. All these actuation, change of shape to engulf the food particle, to make a vacuole, and to move the vacuole, and to navigate the vacuole has to be done by cytoskeleton. Okay, so formation of vacuole is an intelligent function. Sensing of food is an intelligent function. Okay, all these are operated and realized through cytoskeleton. So formation of vacuole. Now, what was taught to us in high school is that okay, the food vacuole is there, it moves inside. Then from inside somewhere there is another vesicle which is filled with lysozyme. And lysozyme is a very corrosive enzyme which comes and it coalesces, it merges into the food vacuole, and thereby it digests the food. Okay, it hydrolyzes the food, right? So lysosome, there are some of these cisternae inside which where lysozyme is stored. Okay, lysozyme is for hydrolyzing, it is for digesting or for dissolving whatever structures are there inside the cell also. Now, lysozyme is said to be the suicide agent of the cell. It could even kill the cell itself, could dissolve the entire cell. Okay. So, obviously, when the enzyme is so corrosive that it can damage the cell itself, okay, there has to be a metered dose of lysosome where it will, according to the proportional to the size of food particle, it has to package that amount of the precise quantity of lysozyme, and it has to come in another vesicle, and the two vesicles have to meet, right? Now if it is a jelly like thing, two vesicles could be moving at random like Brownian motion, and there is a slim probability that they will ever collide. What is the probability of collision like that? But obviously here it is a much more navigated and motivated movement, and in shortest possible path possibly they vacuoles, these vesicles are moving and they are merging, because movement of anything requires much more energy as compared to any other biological processes. So obviously in order to optimize the movement there has to be some efficient navigation, there has to be something has to direct that movement. Okay, so something has to meter the dose of lysozyme that has been packaged, and that vesicle will come and get merged into this food vacuole, and it will dissolve the food, and then it will this the two things will merge and disappear, and the contents will be dissolved in cytosol, and then they will disperse through the cytoplasm, okay, as nutrients, right? That is how it happens. But all these intelligent functions, how the okay, suppose one of the internal cisternae is having lysozyme which has been secreted by the cell, and it has been stored in tanks, in vesicles. Okay, when the food vacuole comes, some cistern gets a signal that yes, this size of food vacuole is there, and in its direction it separates out a chunk of the stored content in the form of a small bubble or a vesicle, and then that bubble moves in the direction of the food vacuole in the shortest possible path. So obviously there is some skeletal member which is passing through both. So it knows this, it knows the cistern also, and depending on the tension which is required to pull this one, because it is propelling, it is propelling the vacuole as well as it is propelling this vesicle of lysosome. Okay, so certain tension is created, something like there is an element which works like a spring in tension, and it maintains the tension and it goes on dissolving by the cytosol, so that the distance between the two vesicles becomes shorter and shorter, and through whatever intermediate things are there, they will give way, and this will come and eventually merge with this somewhere midway, right? So that is what is supposed to happen. Now that could only happen if there are some ropes and which are doing the trick, which are acting like a spring, which are getting dissolved, which are maintaining tension. Okay, so that is the engineering aspect of cytoskeleton. Those are the properties of cytoskeleton, right? So navigation, propulsion of vacuole, optimizing the movement, optimizing the energy required to move these bubbles, etc., inside. Okay, all that is done through cytoskeleton. Okay, now why there are so many parallel cisternae? I'll throw some light on that. These are, of course, the parallel cisternae have been maintained by cytoskeleton in that particular disposition. Now, there is a famous example of insulin. Now, most of the times, whatever proteins get secreted by the cell, they come from certain gene, certain DNA, certain part of DNA, which we call as gene, we choose to call as gene. Fine. Now, in case of insulin, scientists have identified and they are wondering that there are two different genes which are involved in production of insulin structure. So half of the structure come from one gene, other half come from another gene, and the two are quite far apart. They are not neighbors, kind of thing. Okay, so how does it happen? Okay, how would two different genes contribute to make some third product which is not directly as per the genetic code which is there in a sequence, right? Now if you see these cisternae, let us say there is a tank A, there is a tank B, there is a tank C, or maybe from inside out. So this is tank A, this is tank B, tank C, tank D. Okay, this has some chemical which is secreted from some gene. This is another one, some other chemical secreted from some other gene. This is some other fluid secreted from some other gene, and so on. So there are parallel cisternae in Golgi apparatus, and that is why they are made more or less concentric. Now what happens that when a bubble arises from the inner thing, from A to D and outwards to meet the food vacuole, or to be secreted as a bubble by the cell. Okay, like in case of insulin. Okay, the vacuoles are also involved in secretion apart from ingestion. Okay, so some chemicals which are produced can be let out through such things from the surface. Okay, those are the secretions. Like glands do secrete many things. Bone cells do secrete calcium bound by protein matrix, right? So there are secretions in cell. It could be solid secretion, it could be liquid secretion, right? So the cells, the vacuoles, as much they ingest, they can secrete, they can excrete also, as you wish to call it, right? So here, when the bubble is rising from inside, the same bubble passes through the next cistern. It goes through this one, then through this one, then through this one, then comes out. So what you get is a chemical reaction of A, B, C, and D fluid. Okay. Now that explains how insulin comes. Okay. So one cistern populated by one gene product, another one populated by another gene product. The vesicle which is coming out from inside is passing through this as well as this as well as this. So it is a reaction of A, B, C, D, whatever comes on the way, and then it gets secreted. Okay. So the location of cisternae are important. What they produce is important. What they secrete is important. What the cell produces is dependent on that. That's another aspect of structural intelligence of the cell. Now, structural intelligence of the cell is realized by none other than cytoskeleton. Who else can do that? What else can make sure that these vesicles will lie in this particular orientation with respect to each other, and they will come in the way when the vesicles rise from inside and move outward, right? So that is another one. We have spoken about dosing of lysozyme already. Obviously, when the food vacuole is getting pulled by some rope, the tension in the rope tells the other side that what is the mass inside the vacuole, and proportionately from the cisternae, corresponding mass of lysosome gets released. Okay, so that there is no excess lysozyme to dissolve the components of cell itself apart from food. It has to be optimized, right? It has to be a metered dose. So that is realized by cytoskeleton. Also, talking of lysozyme, lysozyme has another important function. It carries out the scavenging of the interior of the cell. Like whatever redundant leftover materials, non-useful materials, damaged materials are there inside the cytoplasm, they have to be cleared up. They have to be hydrolyzed. They have to be recycled. Now, it is the duty of cytoskeleton again to identify such bodies which are disposable. Okay, whatever is abnormal, whatever is excess, whatever is not required, whatever is a fragment, its function is over. All those elements have to be dissolved by hitting it with lysozyme, right? And so that they get hydrolyzed and they get circulated, its elements get circulated inside again as a raw material within cytoplasm. So it is recycling and reutilization in the engineering sense, right? So intracellular scavenging function is a very important function. Scavenging does happen, but who identifies what has to be cleaned, what is normal, what is abnormal, what is required, what is not required? Now that intelligence is realized again through cytoskeleton. What else could do it? Nothing. A jelly cannot do that, right? A jelly cannot pinpoint the location where some scavenging is required, right? Unless it has a map inside, unless there is a structure, unless it knows its coordinates, okay, where the cleaning has to be done, and so on. So scavenging is coordinated and executed through cytoskeleton. Also, you see these holes, the DNAs will come out of it, the genes will make copies of some sequences and they will come out to what is called as endoplasmic reticulum. And through some holes, there will be a transcription and translation, and the sequence will be converted from a genetic sequence to a protein structure. Okay. That is the translation of genetic code into a protein structure. So, which means that these holes, in other words, what are called as mRNAs and rRNAs. mRNAs are messenger RNAs which bring the transcript from inside, from the archive up to here. And here there is a rRNA, ribosomal RNA, which is holding it, running through the tape and converting it into equivalent sequence in terms of amino radicals and making a protein structure, okay, corresponding to the genetic code, right? But when the protein is secreted, it's only the primary structure. It is secreted as a long chain molecule, as a single strand. But how that single strand should be shaped is defined by the interspaces available to that extruded molecule. Okay, it is flexible. So when it is coming out, it has a very limited rigidity. So it is limited stiffness. It will go hit some wall, hit some rope, turn. So it will get confined and restricted by the ropes of cytoskeleton, by the interspaces in the cytoplasm. Okay, so those interspaces, in other words, define the shape of secondary and tertiary and quaternary structure of protein. Primary structure is, of course, a linear sequence, but proteins are anything but a linear sequence when they come anywhere on the site, like on cell membrane. or anywhere else, or as member of cytoskeleton itself. They look like an intertwined rope, possibly not a single strand, but well woven intertwined rope, or even a fabric sometimes could be something like a web, right? Now, all that can happen only if the extrusions which are coming out in the form of proteins, their shape and their navigation is controlled by the interspaces. So it is the role of cytoskeleton to provide the formative shapes and restrict the growth and of structures of protein which are getting extruded from the ribosomal RNAs and rRNAs. Okay, and then of course they have to be navigated and pushed into the site where the protein is supposed to go. Okay, the protein long chain molecules are never produced just to hover around in cytoplasm in a jelly and find their way, because there will be too much of tangling, okay, intertwining. Okay, since they are long chain molecules, they have to be guided. They cannot diffuse through like small molecules, right? So all those functionalities are also carried out by the cytoskeleton. Okay, and correspondingly, I mean, the genetics people say that if you try to hoodwink a gene and force to and change its sequence, you change the genetic code. Still, the cell has intelligence that it does some kind of a proofreading. If the protein produced is not conforming, then it clips it, it hydrolyzes it, breaks it. It does not permit a wrong product to come out. That is the error proofing, fool proofing, whatever you may call. It is being done. That is why the cell systems are so robust. Despite the fluidity and flexibility which they apparently have, they are very, very robust and error-proof systems. Okay, that has been proven statistically. So the proofreading function is obviously done by the shapes, by the interspaces. If some wrong sequence is getting produced, it will not fit into the shape which is meant for it. So then that will restrict the production of that protein, obviously. So proofreading function is also realized through cytoskeleton. Now, the funny part is that most of this research in this cellular biology is done by microbiologists. And what they do is, for every process, they have named some enzyme after the process, and they have left it there. So everything is done by some enzyme, XYZ enzyme, and they leave it there. How that enzyme works is a gray area. Okay. But of course, we can figure out how it could be working, of course, depending on the engineering requirement, depending on the functionality, depending on the optimization, energy efficiency, and these considerations. Right. The biological system, the cell cannot be doing several hit and trial. It's like Brownian movement to make the vesicles. Maybe the vesicles will meet somewhere. It cannot be like that. It has to be well guided, well optimized uh, in terms of energy, in terms of material. Okay, it has to be optimized. The cell is a highly optimized system, right? And who carries out that optimization? Obviously, the cytoskeleton. Okay. So also, when there is a cell division, of course, sometimes the cell divides into two, right? When the cell divides, the cytoskeleton takes the role, and basically, this is how it looks like. All the chromosomes get aligned around the equator of the cell, and two poles appear from which the visible fibers get attached, and they align these chromosomes at the equator of the cell. And after that, the tension comes, something like what I was talking some time back. These are like elastic ropes. They maintain tension and they exert more tension, so it splits in the middle, like here. And one half of chromosome goes in one daughter cell, other half goes in another daughter cell. That's how cell division takes place. And these elements are, for some reason, called as spindle, though they are not rotating spindles in the engineering sense, but they are called as spindles because they carry out some movement within the cell. Okay, but obviously this is pointing something. One thing is that the cytoplasm, the locations of cytoplasm are attached to the genetic material specifically. So every gene is mapped, is hardwired to certain location through cytoskeleton, obviously. That is why a genetic product can be propelled at certain specific location. That is number one. Another thing is these genetics materials archive is identified, and it can be repeatably and intelligently split into two so that they can be duplicated again, and you have two full daughter cells for progeny, right? Now all these mechanisms are clearly realized through these spindles which carry out cell division. Okay, so cell division, mapping of genes with the site within the cell, all these functions are realized through cytoskeleton, the fibrous material within cytoplasm. Okay. So also I have spoken before that the cells, through the surfaces there will be some proteins which will be passing into the next cell. Okay, and they will make tight junction, etc. Sometimes the two cell walls will almost merge with almost zero gap. Okay, and there will be some structural members which will be running from one cell to the adjacent cell, right? So relation with the adjacent cell, relation with the adjacent media outside the cell is also controlled, operated, actuated, sensed through cytoskeleton. How do you like it? So in short, practically all the intelligent aspects of intracellular functionality have one agent which is executing them, that is the cytoskeleton. Okay, so that's all for the day. I hope you have liked the video. If you have liked, please like it, and please look out for our next video in this series. And the next video shall be released very shortly, and that will be dealing with biohardware and what are the wonderful properties which protein structure and macromolecular assemblies offer. Some of it, of course, I have indirectly hinted today, but we'll see in some detail in the next video. Okay, till then, bye bye, and see you in the next video.